Welcome back, everyone. Last week we built the conceptual foundation of hematology. We talked about hematopoiesis is, where blood cells come from, and how stem cells differentiate into myeloid and lymphoid lineages. That week was really about understanding the idea of blood cell development. This week is where we start applying that knowledge. Instead of thinking about hematopoiesis as a diagram in a textbook, we're going to start thinking about what development actually looks like under the microscope. Because in laboratory medicine, morphology is how biology reveals itself to us. As histotechnologists, you may not always be the person making the final diagnosis, but you are absolutely part of the diagnostic chain. The way tissue is prepared, stained, and presented determines whether normal maturation patterns are visible or completely obscured. So this week is really about training your eyes, learning to recognize normal developmental patterns so that later, when we talk about disease, abnormalities actually make sense instead of feeling random or overwhelming. Think of this week as moving from learning the language of hematology to beginning to read it. Let's quickly ground ourselves in where we ended last week. You learned that hematopoiesis is not a static process. It's continuous, highly regulated, and incredibly efficient. Your body is producing millions of blood cells every second, and that production has to stay balanced to maintain normal physiology. We also introduced hematopoietic stem cells, the starting point capable of self-renewal and differentiation into all blood cell types. From there, development branches into myeloid and lymphoid pathways, each producing cells with very different roles in the body. But the most important takeaway from week 1, and the idea that drives this entire course, is that normal development provides the baseline for understanding disease. In hematology, most disorders are not just about too many or too few cells. They are about problems of maturation. Cells develop incorrectly, stop developing, or mature in abnormal ways. So today we pick up right there, moving from understanding developmental pathways conceptually to recognizing how those pathways appear morphologically. Because biology leaves visual clues, and our job is learning how to read them. You'll probably notice a shift in how this week feels compared to last week. There's less abstract theory and more visual interpretation. Instead of memorizing pathways, you'll begin asking questions like, what stage of development am I looking at? Does this cell look immature or mature? Do these cells appear to be developing normally? This is where students often feel a little uncomfortable at first because many cells initially look very similar. That's completely normal. Morphology recognition is a skill that develops through exposure and pattern recognition, not instant memorization. And this shift is intentional. Laboratory professionals don't approach slides by reciting textbook definitions. We observe patterns, compare features, and interpret development logically. You're beginning to move from student learning toward professional thinking, evaluating evidence visually and connecting structure to biologic function. So this week is less about knowing names and more about understanding change over time. This slide might actually be one of the most important ideas in the entire course. Most hematologic diseases are not defined simply by the presence of abnormal cells. They are defined by disrupted maturation. Leukemias, for example, often occur when cells become stuck in immature stages. Anemias can result from ineffective development. Bone marrow disorders frequently reflect imbalance in normal production pathways. But here's the challenge. Abnormal only exists relative to normal. If you don't understand what healthy maturation looks like, everything under the microscope just looks like a collection of slightly different round cells. And that's where students get frustrated. Once you understand normal development patterns, though, abnormalities become obvious. Cells start to look out of sequence, proportions feel wrong, or maturation appears disorganized. And this is where histotechnology plays a critical role. Morphology only reflects biology accurately when specimens are prepared correctly. Fixation, staining, and smear quality directly influence whether chromatin patterns, cytoplasmic features, and maturation stages are visible. And when we say morphology reflects biology, we also mean morphology reflects technique. Your work at the bench directly impacts diagnostic interpretation. And that's why we start with normal, because it gives your eyes a reference point for everything that comes next. All right, now we shift from theory into why this actually matters to you as histotechnologists. Last week we talked about hematopoiesis conceptually, stem cells, differentiation, and developmental pathways. This week we start asking the laboratory question, why do we actually see this in real practice? One of the biggest places is bone marrow biopsies. Bone marrow is essentially the factory floor of blood cell production, and when clinicians suspect systemic disease, leukemia, lymphoma, marrow failure, metastatic cancer, and even chronic infections, this is where they look. And here's the important histotech truth. You are not just processing tissue. You are preserving biological evidence of cell development. Bone marrow morphology gives pathologists clues about systemic disease processes happening throughout the entire body. A subtle fixation issue, overstaining, or poor section quality can completely change how maturation patterns appear under the microscope. So unlike many routine tissues where architecture dominates interpretation, bone marrow interpretation heavily depends on cell detail and maturation recognition, which means processing quality directly affects diagnostic confidence. In other words, bad processing doesn't just make an ugly slide. It can make normal maturation look pathologic or hide real disease. That's why hematopoietic tissues are one of the clearest examples of how histotechnology and diagnosis are inseparable. Let's ground ourselves in the basics. What bone marrow actually is. Bone marrow is the primary hematopoietic organ in adults. It's not passive tissue sitting inside bone. It's one of the most metabolically active environments in the body. Every second, your marrow is producing red blood cells for oxygen transport, white blood cells for immune defense, and platelets for clotting. We're talking billions of cells daily. From a histology standpoint, bone marrow looks chaotic at first glance. Lots of different cell types, different stages of maturation, varying sizes, and scattered fat spaces. But what apparent chaos is actually highly organized biology. Unlike epithelial tissues where structure is obvious, marrow organization is functional rather than architectural. Cells are arranged according to developmental needs and vascular access rather than neat layers. This is why recognizing marrow requires pattern recognition skills instead of memorizing shapes alone. And clinically, continuous production matters because blood cells have short lifespans. Red blood cells live about 120 days. Neutrophils sometimes only hours to days. If marrow stops functioning properly, patients become symptomatic quickly. So when you're preparing marrow slides, you are literally visualizing active physiology, not static anatomy. Now let's talk about something students often memorize but don't always connect to interpretation. Red marrow versus yellow marrow. Red marrow is the active hematopoietic marrow. This is where cell production is happening. Microscopically, you'll see dense cellularity with developing precursors at multiple maturation stages. Yellow marrow, on the other hand, is primarily fatty and less hematopoietically active. It contains more adipocytes and fewer developing cells. Here's where age becomes important. Infants are almost entirely red marrow because rapid growth requires massive blood cell production. As we age, portions of marrow convert to yellow marrow, beginning centrally and moving outward. So adults normally have a mix. And this matters diagnostically because cellularity expectations change with age. A marrow that looks hypocellular in a child might be normal in an older adult. Conversely, a hypercellular marrow in an elderly patient may signal disease. Histotechnologists play a role here too. Proper decalcification and processing are critical because marrow fat and delicate precursor cells are easily distorted. Overprocessing can artificially reduce apparent cellularity. So when we talk about marrow types, we're not just describing anatomy, we're establishing the baseline needed to recognize abnormality. Now we move into one of the most important and honestly coolest concepts in hematopoiesis, the bone marrow microenvironment. Stem cells do not develop in isolation. They live inside what we call a supportive niche, a specialized environment that regulates when cells divide, mature, or remain dormant. This microenvironment includes stromal cells, endothelial cells, macrophages, adipocytes, extracellular matrix components, and vascular structures called sinusoid. Think of sinusoids as controlled exit ramps. Mature cells enter circulation through them once they're ready. Growth factors and cytokines act like biochemical instructions telling cells what lineage to follow and when to mature. Without these signals, normal hematopoiesis cannot occur. From a laboratory perspective, this explains why marrow disease isn't always a problem with the stem cells themselves. Sometimes the environment supporting them is disrupted. Fibrosis, inflammation, or infiltration by tumor cells can alter the niche and change maturation patterns, which is exactly what pathologists evaluate morphologically. And here's the histotech connection again. If fixation collapses sinusoids or processing damages stromal relationships, we lose contextual clues about how cells interact within the niche. So marrow slides aren't just about identifying individual cells, they're about preserving cellular relationships. You're preserving an ecosystem, not just tissue. All right, now we move from what bone marrow is into how it actually works. And this is where things get really interesting. The bone marrow is not just a random collection of developing cells. It is an incredibly organized environment called the hematopoietic niche. Think of it less like a pile of cells and more like a carefully managed neighborhood where every resident has rules, signals, and responsibilities. Stem cells do not mature independently. Their behavior is controlled by surrounding stromal cells, endothelial cells, macrophages, adipocytes, and extracellular matrix components. These supporting cells constantly send biochemical signals that determine whether a hematopoietic stem cell stays dormant, starts dividing, or commits to differentiation. And this matters enormously in pathology. When signaling pathways are disrupted, whether by leukemia, fibrosis, inflammation, or therapy effects, maturation becomes disorganized. Cells may proliferate when they shouldn't or fail to mature properly. From a histotechnology standpoint, this means morphology reflects environment. When you see abnormal maturation patterns on a bone marrow biopsy, you are often seeing a niche problem, not just a cell problem. Another key point, maturation is controlled spatially. Certain signals are stronger near sinusoids, others near bone surfaces. So location inside marrow literally influences cell fate. So when we talk about controlled maturation, we are really talking about a regulated ecosystem, and disease frequently begins when that ecosystem breaks down. Now let's translate the niche concept into something you can visualize under the microscope. Bone marrow is highly organized spatially. Developing cells tend to remain close to stromal support structures early in maturation. These stromal interactions provide survival signals and growth factors necessary for development. As cells mature, they migrate toward vascular sinusoids, or specialized blood vessels within the marrow. These sinusoids function as exit points into circulation. So maturation is not just biological, it's geographic. Immature cells stay protected within the marrow microenvironment. Mature cells move toward the sinusoids, cross the endothelial barrier, and enter peripheral blood. This migration pattern becomes extremely important diagnostically. If mature precursors are seen circulating in peripheral blood, that suggests something abnormal is happening, either excessive production, marrow stress, or malignant disruption of normal retention signals. From a histotechnology perspective, recognizing organization helps interpretation. When preparing marrow biopsies, preservation of architecture matters just as much as cellular detail. Crushing artifact or poor fixation can distort spatial relationships, making interpretation harder for the pathologist. So remember, marrow isn't chaos, it's structured traffic flow. Development, migrate, release. This slide connects directly to diagnostic reasoning. Under normal conditions, immature cells belong in bone marrow, not the bloodstream. Marrow acts as both a production site and a quality control checkpoint. Immature precursors are retained until they reach functional maturity. Only then are they released into circulation, where they can actually perform their physiologic roles. Peripheral blood therefore represents the finished product line. So when we see blasts or early precursors circulating, that immediately raises clinical concern. It may indicate marrow stress, severe infection, recovery after suppression, or hematologic malignancy such as leukemia. Here's where histotechnology plays a quiet but critical role. Bone marrow specimens and peripheral blood smears must be processed differently because diagnostic interpretation depends on preserving subtle morphology differences. Nuclear chromatin patterns, cytoplasmic granules, and maturation stages. If staining quality is inconsistent or smears are poorly prepared, immature cells may be misidentified or missed entirely. So diagnostically, this comparison answers a simple but powerful question. Are cells where they are supposed to be? Normal biology has rules. Disease breaks those rules. Now we arrive at one of the most important interpretive concepts in marrow evaluation, cellularity. Cellularity refers to the ratio of hematopoietic cells to fat within the marrow space, and this ratio changes naturally with age. Young individuals have highly cellular marrow because hematopoietic demand is high. As we age, fatty replacement increases and overall cellularity decreases. A useful rule of thumb you'll hear clinically is expected cellularity is approximately 100 minus the patient age, plus or minus about 10%. So a 30-year-old might normally have around 70% cellular marrow, while an older adult may normally have significantly more fat. This is critical because cellularity alone does not equal disease, so context matters. Hypercellular marrow may indicate leukemia, myeloproliferative disorders, or reactive production. Hypocellular marrow may suggest aplastic anemia, treatment effects, or marrow failure. From the histotechnology side, processing quality directly affects cellularity assessment. Over-decalcification can damage nuclear detail, while poor fixation can cause tissue shrinkage, falsely altering perceived cell density. So when pathologists evaluate cellularity, they are trusting that the histotechnologists preserve the architecture accurately. In other words, cellularity isn't just biology, it's also technique. And this is a perfect example of how laboratory preparation directly influences diagnostic medicine. Now we're going to introduce a concept you will hear constantly in hematology and bone marrow evaluation, the myeloid to erythroid ratio, or the ME ratio. And I want to emphasize something right away. This is conceptual exposure, not memorization week. Nothing is calculating ratios tonight. The ME ratio simply reflects production balance inside the marrow. Think of bone marrow like a manufacturing facility with multiple assembly lines running at the same time. One line produces granulocytes and other myeloid cells, while another produces erythroid precursors that will eventually become red blood cells. The ME ratio tells us whether those production lines are running in proportion to physiologic need. If the body needs oxygen delivery, erythroid production increases. If infection or inflammation is present, myeloid production ramps up. From a histotechnology standpoint, this matters because when we prepare bone marrow aspirates or biopsies, we are preserving the visual evidence of that balance. Poor smear technique, crushing artifact, or even staining or uneven staining can distort perceived cellular proportions, and that can directly impact interpretation. So even though pathologists make the diagnostic call, our technical quality determines whether production balance can be accurately assessed. For now, the takeaway is simple. Bone marrow isn't random cell chaos. It is regulated production, and the ME ratio is one way we measure that regulation. Let's revisit stem cells, because week 1 introduced them conceptually, and now we're placing them into real marrow biology. Hematopoietic stem cells have 3 defining abilities. First, self-renewal. They can make copies of themselves so the system never runs out of starting material. Second, multipotent differentiation. One stem cell can generate multiple blood cell lineages. And third, continuous replenishment, which is critical because blood cells have limited lifespans. Neutrophils survive hours to days, red blood cells last about 120 days, and platelets roughly a week. So marrow is constantly replacing what circulation consumes. The diagram here shows symmetric versus asymmetric division, and this is where biology gets elegant. Sometimes a stem cell divides to expand the stem cell pool. Other times it produces a progenitor cell that commits towards differentiation. From a laboratory's perspective, this explains why we rarely see true stem cells in peripheral blood under normal conditions. They belong in the marrow niche. When we do see immature precursors circulating, that's a biological signal. Something has disrupted normal regulation. So again, morphology reflects biology. When development control changes, cell appearance and location change too. Now we zoom out and reconnect everything to lineage development. This diagram is essentially your roadmap for hematology. All blood cells begin with a hemopoietic stem cell, which branches into 2 major developmental pathways, the myeloid lineage and the lymphoid lineage. The myeloid pathway produces cells you'll frequently recognize morphologically: erythrocytes, neutrophils, eosinophils, basophils, monocytes, megakaryocytes, and ultimately platelets. The lymphoid pathway produces T cells, B cells, NK cells, and plasma cells. Why does this matter for histotechnologists? Because differentiation determines morphology. As cells commit to a lineage, their nuclear structure, chromatin pattern, cytoplasm, and granule formation change in predictable ways. These visual transitions are exactly what we rely on when examining marrow aspirates or correlating marrow findings with peripheral smears. You are not memorizing every branch today. You are learning that hematopoiesis follows organized developmental logic. And once you understand the pathway, abnormal patterns become easier to recognize later when we discuss leukemias and dysplasias. Normal development gives you the map. Disease shows you where the map breaks.
This slide connects biology directly to diagnosis, and honestly, this is one of the most important conceptual ideas in hematology. Disease often isn't about cells appearing out of nowhere. It's about development going wrong. When differentiation is arrested, immature cells accumulate. Instead of progressing through maturation stages, they stop. And that's a hallmark feature of many hematologic malignancies. Alternatively, production may be excessive or insufficient. Too much proliferation leads to crowding and ineffective hematopoiesis. Too little production leads to cytopenias. From a histotechnology perspective, this reinforces why recognizing normal maturation patterns is essential. You cannot identify abnormal unless you understand baseline development. When you prepare marrow sections or aspirate smears, you are preserving a developmental snapshot. Pathologists interpret disease by comparing what they see against expected maturation sequences. So morphology is not just appearance, it is evidence of underlying cellular decision making. In other words, differentiation tells the story of whether hematopoiesis is functioning normally or failing. And as we move forward in this course, nearly every disease we study will come back to one question. Where in development did things go off track? All right, now we're going to talk about something that students often think they need to memorize immediately. And I'm going to save you some stress right now. You do not need to memorize every cytokine name yet. What I want you to understand is the concept. Last week we learned that hematopoiesis doesn't just happen randomly inside the marrow. Stem cells don't wake up one morning and decide, today I feel like becoming a neutrophil. The process is tightly regulated through chemical signaling, and those signals come from growth factors and cytokines. Think of cytokines as the marrow's communication system, essentially text messages between cells saying, we need more red cells, increase neutrophil production, slow down proliferation. For example, erythropoietin, or EPO, signals increased red cell production when oxygen levels drop. GCSF stimulates a granulocyte production, something you'll see clinically when patients receive growth factor therapy after chemotherapy. Thrombopoietin, or TPO, drives platelet formation. From a histotechnology perspective, why do we care? Because these signals directly change what we see morphologically. If cytokine signaling ramps up production, marrow cellularity increases. Lineages expand. Immature precursors may appear more prominently. So when you look at a marrow smear later in this course, you're not just identifying cells, you're seeing the biological response to signaling pathways. Right now, focus on the big idea. Growth factors regulate production balance, not individual cell identity. Later, we'll attach names. Today, we understand the system. Now we move into one of the most important mindset shifts in histotechnology and hematology. Students often think morphology means memorizing pictures. It does not. Morphology means recognizing patterns of biological behavior. This slide shows carcinogenous progression, but the principle applies broadly across laboratory medicine. Cells change gradually, predictably, and logically. Early learners often try to memorize isolated images, and that's exhausting and ineffective. Instead, I want you looking for patterns, progression, direction of change. In hematopoiesis, immature cells don't suddenly become mature overnight. They transition stepwise. Each stage reflects functional readiness. In pathology, disease often represents disruption of that progression. For example, cancer reflects uncontrolled proliferation. Leukemia often represents arrested maturation. Reactive processes show accelerated but orderly development. From the histotechnologist's perspective, your job is not diagnosing, but your slide quality and recognition skills allow pathologists to interpret these patterns accurately. So when you examine marrow smears, think, where is this cell along the developmental journey? Not, what random picture does this match? Morphology is storytelling through structure, and once you learn the story, the cells stop looking random. Now we finally zoom into what actually happens as cells mature, and this is foundational for everything you'll do moving forward. Across nearly all hematopoietic lineages, maturation follows consistent structural rules. First, cell size decreases. Immature precursor cells are large because they are metabolically active and dividing. As cells specialize, they stop dividing and become more efficient, meaning smaller. Second, chromatin condenses. Early cells have open, loose chromatin because genes are actively being transcribed. As maturation progresses, chromatin becomes darker and more clumped. Function becomes specialized rather than flexible. Third, cytoplasm changes. You'll start seeing granules appear in granulocytic cells. Cytoplasmic staining characteristics shift depending on protein production and cellular function. Fourth, nuclear shape evolves. Round nuclei become indented, then segmented, especially in neutrophil development. Here's the key takeaway for laboratory practice. You are not memorizing stages individually yet. You are learning directional change. When looking under the microscope, ask, is the nucleus becoming more condensed? Is the cell getting smaller? Are functional structures appearing? If yes, you're moving towards maturity. Histotechnology fun fact. Fixation and staining quality directly affect how clearly these maturation features appear. Poor smear prep or staining artifacts can completely obscure developmental interpretation. So yes, technique matters clinically. And this slide summarizes the 4 visual anchors you will rely on repeatedly throughout hematology. When students feel overwhelmed identifying cells, it's usually because they're trying to analyze everything at once. Instead, reduce every cell evaluation to 4 questions. 1. Nuclear chromatin. Is it open and delicate or dense and clumped? Open chromatin equals immature. Condensed chromatin equals mature. Number 2, nucleus to cytoplasm ratio, or NC ratio. Immature cells have large nuclei and minimal cytoplasm. As maturation progresses, cytoplasm increases and the nucleus becomes proportionally smaller. This is one of your fastest maturity indicators. Number 3, cytoplasmic granules. Granules represent specialization. Their presence and sometimes color tells you lineage and functional readiness. 4, overall cell size. Again, immature equals large. Mature equals smaller and more efficient. If you remember nothing else today, remember this. Hematology morphology is pattern recognition using consistent structural rules. You are training your brain to recognize biologic maturity visually. And here's the encouraging part. Nobody is good at this immediately. Even experienced laboratorians pause and reassess slides. Skill comes from repeated exposure and structured observation. By the end of this unit, you won't just see purple circles anymore. You'll see developmental biology in motion. All right, this slide is where everything from week 1 starts clicking together. Last week we talked about stem cells, differentiation, and why maturation matters. Now we're translating that biology into something histotechnologists actually do every day. Recognizing whether a cell is immature or mature based purely on morphology. In the laboratory, we rarely get a label telling us what a cell is. Instead, we ask, does this cell look like it's still learning its job or already doing it? Immature cells are essentially trainees. Their main purpose is proliferation, self-renewal, and differentiation. They are metabolically focused on growth and division, not function yet. That's why they tend to have high nucleus to cytoplasm ratios, open delicate chromatin, and round, relatively simple shapes. The nucleus dominates because transcription activity is high, and the cell is actively preparing to become something else. Mature cells, on the other hand, are specialists. They've graduated training. Their morphology reflects function. Red blood cells optimize oxygen transport. Neutrophils optimize mobility and phagocytosis. Lymphocytes optimize immune signaling. You'll notice chromatin becomes dense and condensed. That's because the cell is no longer trying to become something new. It's now focused on doing a specific job efficiently. From a diagnostic standpoint, this distinction is huge. Immature cells belong mostly in bone marrow. When we start seeing large numbers of them circulating in peripheral blood, alarms go off because that often indicates marrow stress, dysregulation, or leukemia. So histotechnology takeaway. We are not just staining cells, we are visually assessing developmental stage, and that stage carries clinical meaning. Now let's zoom in on erythropoiesis, red blood cell production, because this is one of the most elegant examples of controlled cellular maturation in the body. The entire purpose of erythropoiesis is simple: oxygen delivery. But achieving that simple goal requires enormous production capacity. Your body produces roughly 2 million red blood cells per second. Yes, per second, which means bone marrow must operate like a perfectly regulated manufacturing system. Unlike many other cells, red blood cells are highly specialized. Mature erythrocytes lose their nucleus, lose most organelles, and essentially become hemoglobin delivery vehicles. That means every step leading up to maturity is carefully controlled. From a lab perspective, erythropoiesis tells us something critical. Morphology reflects function development. Early erythroid cells look nothing like mature red blood cells because they're still synthesizing proteins, dividing, and building hemoglobin machinery. As maturation progresses, cell size decreases, cytoplasm changes color, nuclear activity shuts down. Clinically, erythropoiesis responds rapidly to physiologic need. Hypoxia stimulates erythropoietin production, signaling marrow to increase red blood cell output. So when we evaluate marrow or blood smears, we are indirectly evaluating oxygen delivery physiology. Histotechnologists are essentially visualizing whether the body's oxygen supply chain is functioning normally. Now we start applying morphology recognition to a specific lineage. Early erythroid precursors are big, and I mean noticeably big compared to mature red blood cells. This surprises students at first because we associate red cells with small size. But remember, immature equals growth phase. These early cells show 3 hallmark features. Number 1, large cell size. They are actively dividing and synthesizing proteins, so they require abundant cytoplasm and nuclear machinery. Number 2, open chromatin. The nucleus appears lighter and more delicate because DNA transcription is extremely active. The cell is producing RNA needed for massive hemoglobin synthesis later. Number 3, basophilic cytoplasm. This blue coloration comes from ribosomal RNA. Lots of protein production equals lots of ribosomes. Here's the histotechnology mindset shift. You are not memorizing names yet. You are learning visual trends. When you see large size, deep basophilia, and open chromatin, your brain should think early developmental stage. Clinically, recognizing early precursors helps identify marrow response. Increased early erythroid cells may indicate regeneration after blood loss or anemia treatment. But excessive immature forms without progression, that suggests ineffective erythropoiesis or marrow pathology. Again, morphology equals physiology. This slide shows one of the most important visual stories in hematology, progressive maturation. Instead of memorizing each step immediately, I want you to watch the pattern. As erythropoiesis progresses, 4 predictable things happen. Again, cell size decreases. Early cells are large because they divide. Mature red blood cells are small because their job is circulation efficiency, not growth. 2, hemoglobin increases. As hemoglobin accumulates, cytoplasm shifts from deep blue to gray to pink. That color change is one of the most reliable maturation clues on the right Giemsa stains. Number 3, the chromatin condenses. Again, the nucleus transitions from open and active to dense and inactive. Eventually, it gets expelled entirely, a rare biological event. And 4th, proliferation decreases. Early stages divide frequently. Late stages stop dividing and focus on function. From a laboratory perspective, this progression matters because normal maturation is orderly. Healthy marrow shows a smooth developmental spectrum. Disease disrupts that order. Leukemias may freeze cells at immature stages. Nutritional deficiencies may slow maturation. Toxic injury may produce abnormal transitions. So when you examine a smear, you're asking, does this look like a normal development timeline? Histotechnologists are essentially reading a biological timeline captured on a glass slide. And once you start recognizing progression instead of memorizing labels, hematology suddenly becomes more intuitive and honestly, a lot more fun. Now we're entering late erythroid development, and this is where erythropoiesis becomes visually and functionally dramatic, because the cell is transitioning from a living, metabolically active precursor into a highly specialized oxygen transport structure. Up to this point, erythroid precursors have been dividing and synthesizing components needed for survival. But now the priority changes completely. The cell's entire purpose becomes maximizing hemoglobin capacity and flexibility for circulation. One of the most unique biological events you will ever see in cell biology happens here: nuclear extrusion. Instead of dying or dividing, the erythroid precursor actively condenses its chromatin, making the nucleus smaller, darker, and metabolically inactive. Eventually, the nucleus is physically pushed out of the cell and removed by macrophages in the bone marrow. Why does this happen? Because a nucleus takes up space and consumes oxygen. Mature red blood cells need every possible micron available for hemoglobin and must remain flexible enough to squeeze through capillaries smaller than their resting diameter. After extrusion, the cell becomes a reticulocyte. Reticulocytes still contain residual ribosomal RNA, which is why special stains reveal a faint network pattern. Clinically, reticulocyte counts are incredibly important because they tell us whether the bone marrow is actively responding to anemia or blood loss. From a histotechnology standpoint, what you should be watching for is a coordinated transformation. Cell size decreases, cytoplasm shifts from blue to pink as hemoglobin accumulates, the chromatin becomes dense and pynotic, and ultimately the nucleus disappears entirely. This is differentiation at its most extreme, a cell sacrificing complexity to achieve efficiency. This slide represents something really important, not just information, but how histotechnologists actually think at the microscope. When experienced laboratorians scan a smear or bone marrow, we are not identifying cells one by one from memory. Instead, we evaluate a set of visual rules that I've talked about that immediately tell us whether a cell is immature, maturing, or fully differentiated. Okay? Again, the first and often fastest assessment is the nuclear to cytoplasmic ratio, or NC ratio. Immature cells prioritize genetic activity, so the nucleus dominates the cell. As maturation progresses, cytoplasm expands because the cell begins producing functional proteins, like hemoglobin in erythroid cells. Next, evaluate the chromatin pattern, as we've discussed previously. Open chromatin looks delicate, almost lace-like. That appearance tells you transcription is active and the cell is still developing. As maturation progresses, chromatin condenses into darker, clumped patterns, signaling reduced gene activity. Then look at cytoplasm color and texture. As we've talked about before, basophilic cytoplasm reflects ribosomal activity and protein synthesis. As hemoglobin accumulates, staining shifts more towards eosinophilia, that familiar pink color you associate with mature erythrocytes. Finally, assess the cell size trends. A common beginner mistake is focusing on absolute size. Experts instead look for progression. Cells should gradually become smaller as they mature. So rather than asking, what cell is this? Train your brain to ask, what developmental direction is this cell moving in? That question turns morphology into a logical process instead of memorization, and that's the skill that makes histotechnologists efficient and accurate. Platelet production is fundamentally different from erythropoiesis or leukocyte development, and understanding that difference helps explain many clinical disorders you'll encounter. Platelets are not complete cells. They are functional fragments produced by megakaryocytes in the bone marrow. The process begins with hematopoietic stem cells differentiating along the myeloid lineage under stimulation from thrombopoietin, a hormone primarily produced by the liver and kidneys. This hormone regulates platelet production based on circulating platelet demand. Here's where things become biologically unusual. Megakaryocytes undergo endomitosis, meaning DNA replicates repeatedly without the cell dividing. Instead of producing daughter cells, the nucleus becomes multilobed and highly polyploid. essentially increasing genetic output capacity within a single enormous cell. Why does this matter? Because one megakaryocyte must manufacture thousands of platelets. The cytoplasm expands dramatically and forms internal membrane systems that outline future platelet boundaries. Long cytoplasmic extensions, called proplatelets, project into marrow sinusoids, where blood flow helps shear off platelet fragments into circulation. From a histotechnology perspective, this explains why platelet disorders often originate from marrow abnormalities rather than peripheral destruction alone. When evaluating marrow slides, you are not just identifying megakaryocytes, you are assessing whether platelet production machinery is functioning appropriately. So platelet biology is less about cell division and more about controlled cellular fragmentation designed for rapid hemostatic response. Megakaryocytes are often students' favorite cells because they are visually unforgettable and diagnostically extremely important. The first thing you notice is size. These cells are massive compared to surrounding marrow elements, sometimes reaching 10 times the diameter of nearby precursor cells. Their sheer scale immediately tells you they play a specialized production role. Next is the multi-lobed nucleus. This structure forms through repeated DNA replication without cytokinesis. The nucleus appears folded or cloud-like rather than neatly segmented. This polyploid state allows the cell to generate enormous amounts of cytoplasmic material needed for platelet formation. Then examine the cytoplasm. Megakaryocyte cytoplasm is abundant and granular, containing platelet-specific proteins and internal demarcation membranes. These membranes essentially pre-package future platelets before release. You may also notice cytoplasmic projections extending toward vascular spaces, early stages of proplatelet formation. Clinically, megakaryocyte morphology provides valuable diagnostic clues. Changes in number, size, or nuclear configuration can signal marrow stress, reactive thrombocytosis, or myeloproliferative disorders. For histotechnologists, this means megakaryocytes are not just landmarks in the marrow, they are indicators of hematopoietic health. When you scan a bone marrow slide, spotting megakaryocytes quickly helps orient you spatially and gives immediate insight into platelet production status. They are essentially the manufacturing hubs of primary hemostasis. All right, now let's talk about why megakaryocytes deserve your attention, because these giant cells are actually one of the easiest orientation landmarks in bone marrow. When you scan a marrow smear or biopsy at low power, everything can initially look crowded and chaotic, but megakaryocytes stand out immediately. They are enormous compared to surrounding hematopoietic cells, sometimes 50 to 100 microns, which makes them excellent visual anchors when you're learning marrow architecture. From a functional standpoint, these cells are platelet factories. Every megakaryocyte produces thousands of platelets through cytoplasmic fragmentation. So when we evaluate megakaryocytes, we're indirectly assessing platelet production. This becomes clinically important very quickly. If megakaryocytes are decreased or absent, platelet counts may drop, increasing bleeding risk. If they're increased, dysplastic, or oddly shaped, that can signal myeloproliferative disorders or marrow stress. As histotechnologists, even though we're not making diagnoses, we absolutely influence diagnostic accuracy. Proper fixation, smear quality, and staining allow pathologists to evaluate nuclear lobulation and cytoplasmic detail, features that determine whether platelet production is normal or abnormal. So think of megakaryocytes as both a landmark and a functional indicator. When you see them, you're not just spotting a big cell. You're seeing real-time evidence of how well the marrow is maintaining hemostasis. Now we shift from platelet production to another major arm of hematopoiesis, granulocytes. Granulocytes are key players in innate immunity, meaning they provide rapid, nonspecific defense against infection. Unlike adaptive immune cells that require activation and memory, granulocytes respond immediately, often within minutes to hours. The three main granulocytes you'll encounter are neutrophils, eosinophils, and basophils, each defined by their cytoplasmic granules and functional specialization. Neutrophils are the frontline responders. They migrate to infection sites, perform phagocytosis, and release enzymes that destroy bacteria. When you see elevated neutrophils clinically, think acute bacterial infection or inflammation. Eosinophils are more involved in parasitic defense and allergic responses. Their bright eosinophilic granules contain proteins toxic to parasites, but also contribute to allergic tissue damage. Basophils, although rare, participate in inflammatory signaling and histamine release, linking hematologic and allergic processes. From a histotechnology perspective, granule visualization depends heavily on staining quality. Romanowski-type stains must be balanced correctly, otherwise granules may appear washed out or overly dense, making identification difficult. So when students look at these cells, I want you thinking beyond morphology. You're seeing immune readiness. Each granule represents stored biochemical weaponry, and proper lab technique ensures those diagnostic clues remain visible. Now we begin the granulocytic maturation sequence within the myeloblast, the earliest recognizable precursor in the myeloid lineage. The myeloblast is essentially a blueprint cell. Its primary job is proliferation and differentiation rather than function. Because of this, most of the cell's volume is devoted to the nucleus. Notice the defining features: large nucleus, fine delicate chromatin, prominent nucleoli, very high nuclear to cytoplasmic ratio, and minimal or absent granules. That open chromatin appearance tells us the DNA is transcriptionally active. The cell is rapidly producing proteins needed for future development. Clinically, recognizing myeloblasts is extremely important. Increased blasts in marrow or peripheral blood may indicate acute leukemia. Even small differences in chromatin texture or nucleoli visibility can influence interpretation. For histotechnologists, staining precision matters enormously here. Overstaining can obscure nucleoli, understaining can make chromatin look artificially immature. Either issue can complicate evaluation. So when you see a myeloblast, think of it as a stem-like expansion phase, a cell preparing for specialization but not yet equipped for immune defense. This is the beginning of a maturation journey where structure gradually shifts towards function. Now we move into one of the most visually important transitions in granulocyte development, the progression from promyelocyte to myelocyte. This is where cells begin acquiring the machinery required for immune activity. In the promyelocyte stage, primary or eosinophilic granules first appear. These granules contain enzymes like myeloperoxidase and lysosomal proteins that will later help destroy pathogens. The cytoplasm becomes more prominent, and although the nucleus is still relatively large, chromatin begins to condense. As the cell transitions into the myelocyte stage, several key changes occur. The nucleus becomes more round or oval, the chromatin becomes coarser, and nucleoli disappear. Specific secondary granules begin forming. This is a critical moment because lineage commitment becomes visible. Depending on granule characteristics, the cell begins differentiating towards neutrophil, eosinophil, or basophil pathways. From a laboratory standpoint, this stage is where staining quality really shows its value. Granules must be distinguishable without obscuring nuclear detail. If stain balance is off, students and even experienced reviewers may struggle to distinguish stages. Conceptually, this phase represents the shift from proliferation towards specialization. The cell is no longer just multiplying. It is preparing for a defined immune role. So when you compare promyelocytes and myelocytes, focus on granules and chromatin. Those two features tell the entire story of maturation progression. Now we're moving into the stage where granulocytes start looking much more like the functional cells you'll see in peripheral blood. The transition from metamyelocyte to band cell represents one of the most important recognition shifts for histotechnologists because nuclear shape becomes the primary diagnostic clue. The metamyelocyte is defined by its characteristic kidney bean or indented nucleus. The key detail here is that the indentation is present but does not exceed half the nuclear diameter. Chromatin at this stage is already becoming more condensed, signaling decreasing proliferation activity and increasing maturation. As development continues, the nucleus elongates further into the band form, sometimes called the juvenile neutrophil. In band cells, the indentation deepens so that the nucleus forms a curved or horseshoe shape. Cytoplasm increases slightly and secondary granules are well established, meaning the cell is nearly ready for immune function. From a laboratory perspective, recognizing this transition matters because increased band cells in peripheral blood, what clinicians call a left shift, often indicates infection, inflammation, or marrow stimulation. For histotechnologists reviewing marrow smears, this stage helps confirm normal maturation progression. You should see a smooth continuum rather than abrupt jumps between stages. When that orderly progression breaks down, that's when pathology becomes a concern. Here we arrive at fully mature neutrophil, the endpoint of granulocytic maturation and one of the most common leukocytes you'll encounter in peripheral blood smears. The defining feature is the segmented nucleus, typically divided into 3 to 5 lobes connected by thin chromatin strands. This segmentation is not random. It increases nuclear flexibility, allowing neutrophils to migrate through vessel walls and into tissues during infection. At this stage, the cell is no longer focused on division. Instead, it is completely specialized for innate immune defense. Cytoplasm contains abundant specific granules filled with antimicrobial enzymes and reactive molecules used to destroy pathogens. From a histotechnology standpoint, mature neutrophils are excellent internal quality indicators. When staining quality is optimal, you should clearly see nuclear segmentation, crisp chromatin detail, and subtle cytoplasmic granulation. Poor fixation or staining often obscures these features first. Clinically, neutrophils respond rapidly to bacterial infection, trauma, and inflammation. Increased numbers suggest acute response, while abnormal nuclear shapes, such as hypersegmentation or hyposegmentation, can indicate nutritional deficiencies or marrow disorders. So when you recognize a mature neutrophil, you're not just identifying a cell, you're confirming successful completion of an entire maturation program that began back at the myeloblast stage. This slide brings everything together by emphasizing the recognition pattern of granulocytic maturation. Rather than memorizing isolated cell types, experienced histotechnologists learn to recognize predictable developmental trends. First, again, cell size gradually decreases as maturation progresses. Early precursors are large and metabolically active, while mature cells become more compact and specialized. Second, the nucleus transforms dramatically. We move from round nuclei with delicate chromatin and blasts to indented forms and metamyelocytes to band shapes and finally segmented nuclei in mature neutrophils. Chromatin simultaneously becomes darker and more condensed, reflecting reduced transcriptional activity. Third, the nuclear to cytoplasmic ratio decreases. Early cells appear nucleus heavy, but as differentiation occurs, cytoplasm expands and develops functional granules. And finally, granule development follows a predictable order. Primary azurophilic granules appear first, followed by secondary specific granules that define granulocyte function. For histotechnologists, pattern recognition is essential. You are not evaluating one cell in isolation, you are evaluating whether the entire marrow environment demonstrates orderly maturation. Normal hematopoiesis looks like a gradient, not a collection of unrelated cells. When that pattern is disrupted, too many blasts, missing stages, or abnormal morphology, it signals potential hematologic disease. Now we shift from the myeloid lineage to the lymphoid lineage, which follows a very different differential philosophy. Unlike granulocytes and erythrocytes, which primarily serve immediate physiologic functions, lymphoid cells are responsible for adaptive immunity, meaning targeted, memory-based immune responses. Lymphoid development begins with the common lymphoid progenitor derived from the hematopoietic stem cell. From here, cells commit to either the B cell lineage or the T cell lineage. B cells typically mature within bone marrow and later differentiate into plasma cells responsible for antibody production. T cells migrate to the thymus, where they undergo specialized education to recognize self versus non-self. Morphologically, lymphoid maturation is less visually dramatic than granulocytic maturation. Nuclear segmentation does not occur, and cells remain relatively small with dense chromatin as they mature. Because of this subtlety, lymphoid evaluation relies more heavily on nuclear texture, cytoplasmic characteristics, and clinical context. For histotechnologists, this means lymphoid assessment requires careful attention to population patterns rather than obvious structural transitions. Small changes in chromatin appearance or cell uniformity may carry significant diagnostic importance. Understanding this distinction prepares you for recognized lymphoid disorders, where abnormal proliferation may look deceptively uniform compared to the diverse appearance of myeloid maturation. All right, now we're shifting into the earliest recognizable lymphoid precursor, the lymphoblast. And this is where students often get tripped up because morphologically, blasts across lineages can look deceptively similar at first glance. What makes a lymphoblast recognizable is primarily its nuclear dominance. Notice the extremely high nuclear to cytoplasmic ratio. The nucleus takes up most of the cell, leaving only a thin rim of cytoplasm. From a histotechnology perspective, this matters because fixation and staining quality directly influence whether you can actually appreciate these subtle features. The chromatin here is described as fine or delicate, sometimes called lacy. That appearance reflects active transcription. This cell is metabolically busy and still developing. You may also see prominent nucleoli, which signal active chromosomal production. In practice, nucleoli visibility can change depending on staining intensity and smear thickness, so preparation quality really matters. One thing I always emphasize, blasts are not identified by one feature alone. Recognition comes from a pattern. Large size relative to red blood cells, smooth nuclear contours, fine chromatin, and minimal cytoplasm. Clinically recognizing lymphoblasts becomes critical because increased numbers in peripheral blood or marrow immediately raise concern for acute leukemias, particularly ALL. So what looks like a simple morphology exercise is actually the first step in detecting serious disease. As histotechnologists, your job is ensuring the morphology is preserved well enough that someone downstream can make that call confidently. Now compare that immature lymphoblast to the mature lymphocyte, and you'll immediately notice how dramatic morphology changes with maturation. Mature lymphocytes are smaller and much more compact. The chromatin becomes dense and clumped, which reflects reduced transcriptional activity compared to blasts. Instead of that open, delicate nuclear pattern, you now see dark staining and a more condensed appearance. The cytoplasm is scant, often just a thin blue rim around the nucleus. Under the microscope, beginners sometimes mistake these for artifacts or even platelets if they're scanning too quickly. That's why size comparison with surrounding red blood cells becomes one of your best orientation tools. You'll also notice variability. Small, medium, and large lymphocytes exist. That variation is normal and reflects functional diversity within adaptive immunity. Not every lymphocyte should look identical, and recognizing physiologic variation prevents overcalling pathology. For a histotechnology standpoint, smear quality plays a huge role here. Overstaining can obscure chromatin texture, while poor fixation may cause nuclear smudging, making lymphocytes difficult to distinguish from degenerating cells. Functionally, these are now fully operational immune cells, so B cells, T cells, and NK cells, circulating and ready to respond to antigen exposure. Morphology reflects that readiness, compact, efficient, and specialized rather than developmental. So what we're seeing is maturation translating directly into structural efficiency. This slide is where everything starts coming together conceptually. Both myeloid and lymphoid cells originate from the same hematopoietic stem cell, but their maturation pathways, and therefore their morphology, diverge significantly. Myeloid cells generally become larger, develop more cytoplasm, and acquire visible granules as they mature. Granules are functional structures tied to innate immune responses, so morphology reflects physiology. When you see granules, segmentation, or abundant cytoplasm, you're usually thinking myeloid lineage. Lymphoid cells, on the other hand, remain comparatively smaller with sparse cytoplasm and maintain nuclear prominence. Their function relies more on receptor specificity and signaling rather than granular enzymatic activity, and their morphology mirrors that. This comparison becomes incredibly important diagnostically. Many hematologic diseases are classified based on lineage, myeloid versus lymphoid, and morphology is often the first clue before flow cytometry or molecular testing even begins. For histotechnologists, preparation artifacts can actually mimic lineage differences. Poor staining may hide granules, while thick smears can distort nuclear appearance. That means technical quality directly influences diagnostic interpretation. So this slide isn't just academic comparison. It's training your brain to recognize lineage patterns quickly during slide review. You're learning to ask, does this cell behave like a granulocyte lineage cell or a lymphoid immune cell? This final concept slide answers the big question students often have. Why do we spend so much time memorizing cell appearance? Because in hematology and histotechnology, diagnosis begins with morphology. Patterns of maturation tell us whether hematopoiesis is proceeding normally or whether something has gone wrong. When maturation stops prematurely, you see blasts accumulate. When maturation accelerates, you may see increased bands or reactive changes. Morphology becomes a visual record of underlying physiology. Every structural feature you see, chromatin density, nuclear segmentation, or cytoplasmic granules, reflects cellular function. Structure and physiology are inseparable. And this is where histotechnology plays a critical role. If fixation is delayed, staining inconsistent, or smears poorly prepared, morphology becomes unreliable. That doesn't just affect aesthetics, it affects clinical decisions. A pathologist cannot interpret what cannot be clearly seen. So when we say morphology matters, we're really saying your technical work directly impacts patient diagnosis. The slide you prepare determines whether subtle maturation abnormalities are detectable. The takeaway here is that morphology isn't memorization for its own sake. It's pattern recognition tied to biology, clinical reasoning, and ultimately patient care. And honestly, once you start recognizing these patterns, peripheral smears stop looking random purple dots and start telling a story. All right, now we shift from what cells are to something just as important, how we allow them to be seen correctly. Because here's the truth every histotechnologist learns very quickly. Morphology only matters if the specimen quality supports it. All the beautiful maturation patterns we just learned, myeloblast to neutrophil, lymphoblast to lymphocyte, completely fall apart if specimen handling is poor. Let's break down why each step matters. First, fixation. Fixation preserves cellular architecture at the moment of collection. If fixation is delayed or inadequate, cells begin degenerating almost immediately. Nuclei swell, chromatin blurs, cytoplasm vacuolates. and suddenly immature cells can look reactive or abnormal cells can look deceptively normal. And in hematology, that can mean the difference between recognizing leukemia versus missing it entirely. Next, slide preparation or smear preparation. This is where technique becomes art. Too thick, and cells overlap and nuclear detail disappears. Too thin, cells rupture or distort. Bad feathered edge, you lose your optimal evaluation zone. Histotechnologists and laboratory professionals don't just prepare slides. We create the environment where morphology becomes interpretable. Finally, staining quality. Romanowski stains rely on precise pH balance and timing. Slight variation changes color intensity, which directly affects how we interpret granules, chromatin density, and cytoplasmic features. And remember, many diagnostic decisions rely on subtle color changes. So the takeaway here is simple but powerful. We are not passive observers of morphology. We are active guardians of diagnostic accuracy through specimen quality. No pressure, but also, yes, pressure. Let's pull everything together because this entire lecture really comes down to three core ideas. First, bone marrow is the site of development. Every mature blood cell we see circulating began as a stem cell undergoing highly regulated differentiation in the marrow. What looks random under the microscope is actually a carefully orchestrated biological progression. When you understand where cells come from, their appearance starts making logical sense instead of feeling like memorization. Second, maturation follows predictable patterns. Across the myeloid lineage, we saw consistent trends. Cell size decreases, nuclear chromatin condenses, nucleus shape changes to recognizable stages, and granules appear and specialize. The nuclear to cytoplasmic ratio shifts. These are not isolated facts, they are patterns. And histotechnology is fundamentally pattern recognition science. Once students stop trying to memorize individual cells and start recognizing progression, everything becomes easier and honestly, much more fun. Third, and this might be the most important concept, normal recognition must come before disease understanding. You cannot identify abnormal maturation if you don't deeply understand normal maturation first. Pathology is deviation from baseline. So every time you study normal morphology, you're actually training your diagnostic brain for future clinical interpretation. This is why foundational morphology matters so much in laboratory education. You're not just learning cells, you're learning how to recognize when biology breaks its own rules. So as we wrap up today, I want to zoom out for just a moment. What we covered wasn't just hematopoiesis or cell identification. You learned how laboratory professionals read a story written in morphology. Every smear represents a dynamic system. Bone marrow production, immune response, physiologic stress, disease processes unfolding in real time. And unlike many areas of healthcare, laboratory professionals often make these observations before symptoms fully explain what's happening. That's powerful. As histotechnologists, your work directly determines whether morphology can be trusted, whether clinicians receive clear information, and ultimately whether patients receive accurate diagnoses. So when you sit at a microscope, you're not just identifying cells, you're asking, is maturation appropriate? Does this pattern make physiological sense? Is something developing too early, too late, or not at all? In that mindset, curiosity paired with technical precision is what transforms slide review into professional expertise. If there's one thing I want you to remember leaving this lecture, it's this. Morphology is not memorization. It is interpretation grounded in biology. And the more comfortable you become recognizing normal patterns, the more confidently you'll recognize disease when it appears. All right, that officially brings us to the end of this section. Next time, we'll start applying this knowledge to clinical correlations, where things get really interesting. Nice work today.